Lyme disease, the bacterial infection transmitted by ticks, is the most common vector-borne disease in the U.S., with nearly 500,000 people diagnosed and treated each year, according to the CDC. It can often be resolved with a short course of antibiotics, but in other cases, symptoms such as fatigue, inflammation, pain, sleep problems, depressed mood, and clouded thinking can persist for months. Our guest today, Dr. Brandon Jutras, recently published 2 pivotal studies in Science Translational Medicine about Lyme disease that provide important insights into what may cause persistent symptoms and points to some promising new directions for both diagnosis and therapeutic intervention. He is an associate professor of microbiology immunology at Feinberg and has been studying Lyme disease for more than 15 years. Welcome to the show.
Thanks so much for having me.
Well, let's talk about Lyme disease a little bit. Why is this such a challenging infection to study, diagnose, and treat compared to other bacterial diseases?
First and foremost, of course, it's a vector-borne disease, meaning that it is being transmitted from infected ticks to human or many other animal reservoirs in nature. And when that bacterium that lives inside the gut of the tick is transmitted through the salivary glands, it quickly disseminates throughout the body. And so it doesn't stay in one place, and it very quickly reaches distal tissues in a variety of different hosts. But importantly, it never really gets up to very high densities as well, meaning that you don't have a huge population of bacteria in any one environment. It is also kind of a stealth pathogen, meaning that in essence, it's not trying to make us sick, it's trying to avoid in the innate and adaptive immune systems of humans to basically establish infection. And so the challenges associated with diagnosing Lyme disease are simply that it doesn't get up to very high densities, that it moves extremely quickly and moves throughout the body. And it's extremely challenging to culture the bacterium in a laboratory setting or in a clinical setting. Those are kind of the hallmarks that we think of when we think of diagnosing a bacterial infection, is basically being able to isolate or take that organism and propagate in a laboratory outside of the animal or the human. And so all of those challenges are contributing to our lack of a strong, direct diagnostic test. I would also argue that the material that we use to propagate the bacteria in a laboratory setting or in a clinical setting is both exceptionally expensive and exceptionally complicated to make. So there's a lot of batch-to-batch variability. And so I would say that there's a myriad of factors that are all associated with the kind of diagnostic challenge.
You have been studying this disease since your graduate training years, and you recently moved your lab to Northwestern from Virginia Tech. Tell me, way back when, what sparked your interest in this disease, and how is it being studied in your lab today?
I started doing research as an undergraduate student in a variety of different areas, and in physical chemistry and molecular genetics, and I was interested in spiders and spider silk and different types of arthropods and some of their kind of unusual properties and features, and became interested in ticks and started to study a little bit of tick biology and tick physiology, but then started to become more interested in the things that were actually causing the disease. Again, the ticks are just the vehicle or the vector. The diseases that they're causing and carrying are within them, and those are largely bacterial infections and viral infections. And so I became more interested in the microbes and the kind of business side of the infection or the organisms that were actually causing disease, and less so on the vehicles that were transmitting it. And so yes, I did my graduate work and my postdoctoral work in understanding the bacterium that causes Lyme disease, because it's exceptionally unique and unlike virtually any other bacterium that I'm aware of. And so that is kind of the approach, and the types of things that we do in the laboratory is we're asking fundamental questions about this very unusual organism, and then we let the answers dictate our approach to understanding the disease, so understanding how we're getting sick and why we're getting sick, but then also how we can treat that, how we can prevent that. So we're very interested in, again, understanding basic biological processes associated with the microbe, and we're asking those types of questions in the space of understanding the disease, understanding how we can treat it and treat it in different ways by using some of these unusual properties, and then also leveraging all of this information to develop biomarker or diagnostic-based approaches that are very rapid, very simple, and very accurate.
And you recently moved to Northwestern. Can you tell me how your move to Northwestern is helping you reach some of these goals?
We're thrilled to be here. Yeah, we've been here about a year and a half now. Being in a medical setting, being here at Feinberg gives me access to world-class infectious disease physicians. I actually just returned from doing grand rounds and presenting a seminar to all the infectious disease folks to update them on what we're doing. So being able to have those collegial interactions is absolutely fantastic. It also will hopefully in the future be giving us access to more clinical samples so that we can take our basic science understanding and our basic science approaches and then translate those into real deliverables for patients. And being across the road literally from these physicians and from potentially these types of samples is a real game changer for us. And then Northwestern is just a fabulous environment to do research. And that really opens doors as far as being able to recruit scientists that I can train and work with and get them excited about what we're doing to drive the research forward. Because really the laboratory, that the group of fantastic young scientists we have, they're the lifeblood. They're everything associated with our laboratory. And so to being able to recruit and retain the top talent junior scientists and being able to hopefully help and shape their careers, that's also a long-term passion and goal of mine.
Right now, if someone is diagnosed with Lyme disease, how do they get that diagnosis? And if they have these persistent symptoms, what is the course of action right now?
If you get bit by a tick, of course, not all ticks carry Lyme disease. Only some species of ticks carry Lyme disease. And then, of course, within those species of ticks or that particular group of ticks, obviously in the United States, we're talking about the black-legged tick or Ixodes scapularis. Only a subset of them are actually carrying the bacterial infection inside of them. However, if you are bitten by a tick and you suspect that it is the tick that carries Lyme disease and you go to see your primary care physician, there's a couple different options. At this point, they're looking for a bullseye rash because that's a hallmark of an acute stage of Lyme disease infection. So they're looking for a bullseye rash. If they don't see that, they will ask a series of questions in terms of symptomology, and they may prescribe post-exposure prophylactic treatment, meaning they do not have a diagnostic in hand. However, the circumstances suggest that Lyme disease is possible. And then they may prescribe doxycycline, either a single dose or there are a couple other kind of regimen variations thereof. However, it's unlikely that they'll even order a serology or a blood-based test at that point because it will be negative. The problem with the current diagnostic is that it's a serology or a serum-based test, and they're detecting your response to the infection, not the infection, meaning it's your antibody titers or the levels of antibody that you possess against particular factors and proteins that the bacterium is making, meaning that takes time. Okay, so it takes anywhere between 3 and 6 weeks. It also means that it requires a functional immune system. So if you're immunocompromised, if you're having some form of therapy that temporarily makes you immunocompromised, it's highly unlikely that you'll develop enough antibody that will be detected. And then that test that is being ordered, it's a 2-tier test, and you have to get through phase 1 to get to phase 2. And so the whole process it can take up to 2 months. And there are a lot of questions and a lot of uncertainties when it comes to Lyme disease, but there's one thing that I believe we can all agree upon, both as basic scientists and physician scientists and infectious disease physicians, that the most important indicator of outcome is how promptly and properly you are treated. So here you are in this window of 6 to 8 weeks, which is the critical treatment window, and you don't have a diagnosis yet.
Yeah. But the course of antibiotics that is often prescribed, that can help to clear the infection?
Yeah. So most often oral doxycycline is prescribed. And most often, if it's prescribed early enough and the course of the antibiotic therapy is followed, the doxycycline tends to work quite well. There are a subpopulation of people that will not be prescribed doxycycline for a variety of reasons. And then the dose that is used to treat Lyme disease, specifically the dose of doxycycline that is being used, is a very high dose for an extended period of time. And so what that means is that it's not just attacking the bacterium that causes Lyme disease, it's attacking all the bacteria in your body. And we know that the human microbiome is an exceptionally important part of our body's function. There are more bacterial cells in and on your body than there are your own cells. And they perform essential functions in basically virtually every single physiological process you can think of. Metabolism, your immune system, your cognition, brain function, everything, the microbiome is playing a role. And so if you're getting this extremely high course of doxycycline for an extended period of time, you are largely wiping out a good portion of those essential microbes.
So there's the problem that the treatment for Lyme disease can be incredibly damaging to the microbiome. There's also the fact that some people have a very long, drawn-out battle with Lyme disease that can be very debilitating. Can you talk about that aspect?
It's finally starting to become more well accepted is the idea that there are instances in which doxycycline just doesn't work. And this is this group of patients that we refer to as post-treatment Lyme disease patients, and they develop post-treatment Lyme disease syndrome or symptoms. And so again, it's a subset. It looks like it's anywhere between about 10 and 15% of patients that have a documented case of Lyme disease. So they went through that grueling diagnostic process and it's clear that they have Lyme disease. It's clear that they've been treated with doxycycline and had the proper course, and yet they continue to experience symptoms. And that is another very important and big area of research in our lab is understanding what is happening with these patients. Why is it that they've been treated and they continue to experience symptoms? It's possible that they continue to have a very low level infection. Some folks are under the impression that's the case. We think of it slightly differently. I don't think that's necessarily incorrect. I think that there is likely a subset of patients that probably do have a very low level infection, but to continue to experience these kind of dramatic symptoms, especially we'll kind of uncouple the two, but if we think of something like Lyme arthritis, when you have this massively swollen joint and debilitated syndrome that you can barely walk, for that to be caused by an infection that we cannot detect, it seems like there's other things going on. And that's one of the areas that we've been interested in now for a while. And it stemmed from research during my postdoc that we've now been following in my lab ever since. And it's the idea that during the course of the infection, the bacterium left something behind. And it is this molecule that's creating maladaptive, inappropriate inflammatory response. And that some of these different types of sequelae associated with Lyme disease may not necessarily be entirely an active infection, but really how we responded to the infection and how we continue to respond to the infection. And I think some of these areas of research, often referred to as post-acute infectious syndromes, more kind of broadly speaking, are starting to really gain attention with long COVID. It's very clear that there's something happening here. The molecular mechanisms and the molecular underpinning of what is occurring is still very much an active area of research. But we think that there might be some parallels in the idea that your body may continue to respond to a molecule that the virus or the bacterium, in the case of Lyme disease, left behind that is continually driving low-level inflammation. And it's that uncontrolled response that really is the problem.
What is that molecule called?
So in the context of Lyme disease, it appears as though it's the peptidoglycan of Borrelia burgdorferi. And we can detect this molecule in the synovial fluid or in the liquid that bathes your articulating surfaces, your joints, like your knee. We can find this molecule in the synovial fluid of Lyme arthritis patients in these massively swollen knees weeks and months after they've been treated with antibiotics, but continue to have these symptoms. And this molecule is known to be inflammatory. So if we take that molecule and we inject it in, say, an experimental animal model system, those mice develop arthritis almost immediately. Almost all bacteria have peptidoglycan. So it's not unique to Lyme disease. Virtually all bacteria have peptidoglycan. It's just a big molecular bag that basically protects the inside of the cell. But it turns out the bacterium that causes Lyme disease, the chemical properties of their peptidoglycan are fundamentally different from all other bacteria. And it's those chemical properties that are driving the persistence, at least from what we can tell in experimental animal models and then follow-up studies on human patient samples.
Because these people can suffer, like you said, weeks, months, years after an infection?
Yes, it really does appear. In the context of post-treatment Lyme disease syndrome, these patients, it can be 6 months, 1 year, 2 years. Yes, it's really dramatic. We're still trying to understand that in the context of the peptidoglycan persistence, if it's driving that. But what we have, I think, pretty well demonstrated is that it's clearly playing an important role in arthritis.
You recently had two preclinical studies. They were both published in the April 23rd, 2025 issue of Science Translational Medicine that demonstrate promising implications for improved Lyme disease treatment. So let's start with the study where your team screened nearly 500 drugs to identify piperacillin as a promising candidate to treat Lyme disease. Tell me about this screening process and why piperacillin stood out.
It was a grueling screening process that took a few years and a lot of really fantastic, dedicated junior scientists that really drove that science forward, including the first author, Megan Gabby, who's now in medical school at Virginia Tech. But what Megan did was we took advantage of another unusual feature of Lyme disease, and this is one of these kind of consistent themes when it comes to Lyme disease and Borrelia burgdorferi, is that they're highly unusual organisms. One of the other weird things about them is the way in which they grow. They replicate themselves by growing in these kind of coordinated regions. But the point is that we basically used a quantitative microscopy approach so we could watch this replication process happen in real time and then add tiny amounts of all these different drugs to see at a very quantitative level how the organism was responding to nearly 500 different antibiotics. And the key was that we used all of these antibiotics at a very low concentration with the idea that something that is going to specifically interfere with this process, if it does so very well, it will at a very low concentration. We found a subset of drugs that worked effectively in the laboratory at killing Lyme disease, killing Borrelia burgdorferi. But then what we did is we counter-screened our subset against more generic bacteria to try and weed out those ones that may have kind of off-target, non-specific effects. And again, with the idea of finding a compound that specifically interferes with this weird way that they grow. And in doing so, piperacillin kind of fell out of this screen. And then we went on to identify the target inside of the bacterial cell that is why this antibiotic, this beta-lactam, is so good at basically killing Borrelia burgdorferi. It just turns out that this molecule has a very high affinity and specificity for one enzyme in the Borrelia burgdorferi genome. And that particular enzyme is essential for them to survive. And then we basically took that on and tested it in an animal model as well. A laboratory-based infection where we're using mice, which are a natural reservoir of Lyme disease, to understand the efficacy of this therapeutic option, and found that it works as well as doxycycline, but at 100 times less the dose.
There with the microbiome, then, the gut microbiome.
Right, yeah. So at that very, very low concentration, it's not impacting any other organisms, including all of the microorganisms in your gut, or in the mouse gut at least, because this is, of course, preclinical. But it is working at totally eradicating Lyme disease as far as we can tell. And again, it can be administered at such a low dose that it doesn't impact anything else.
Why is that combination of potency and microbiome protection so important when we're talking about the future of Lyme disease treatment?
We obviously want something that's going to 100% clear the bacteria that we're interested in clearing. And the goal of basically any antimicrobial is for just that, for it to work at effectively killing 100% of viable bacteria in an infection, and then hopefully not kill anything else. And so that combination of the two is really the standard that we hope we can do with any bacterial infection. Frankly, I'm not aware of any where we have anything that's that specific. It just turns out that they don't wipe out the microbiome quite like doxycycline does.
Doxycycline, I'm wondering, how did that become sort of the treatment option for Lyme disease? Do you know what the history is there?
Oh gosh, the history goes back a ways. Doxycycline is primarily known as a effective and potent antibiotic at treating bacteria that are not inside eukaryotic cells. So we refer to these as extracellular pathogens. So bacteria that exist in between systems, not inside cells. We have basically two flavors of pathogens. You have bacteria that are intracellular pathogens, so they live inside eukaryotic cells. They cause disease inside eukaryotic cells, either by manipulating those eukaryotic cells or by killing them. And then you have a subset of pathogens that are not intracellular and they're extracellular, and they live basically entirely outside of cells. And it's well known that doxycycline works very well at extracellular pathogens. And Borrelia burgdorferi is largely an extracellular pathogen. And so that was the kind of simple, I think, conclusion of why doxycycline makes the most sense, is because it is known to be quite potent and it is known to largely target extracellular bacteria.
When folks are prescribed this medication currently, is there anything that's being done to help rebuild the gut microbiome? Is there guidance that's given to patients?
I can't speak to how every physician talks to their patients, but from what I have seen and the guidelines that I've read, and it's very common for a probiotic to be encouraged, certainly to eat lots of yogurt, live cultured yogurt,
Right.
and those types
Okay.
of things. That's very common for physicians to make sure that they mention that because the side effects for doxy can be significant with the microbiome. But another aspect of doxycycline that is becoming increasingly well recognized is that it does impact eukaryotic cells too. So it not only does it impact the microbiome, but long courses of doxycycline at particular doses can have impacts on how our cells function as well.
Okay, so again, underscoring the need for a different treatment, especially for these Lyme disease patients. The second Science Translational Medicine paper looks at why some patients continue to experience these symptoms after treatment, and these symptoms really mimic a chronic illness. Tell me more about that. What did you discover?
Yeah, so as I mentioned earlier, we had been finding consistently that we could detect the peptidoglycan in the patients or the synovial fluid patients of Lyme arthritis knee joints, right? Suggesting that this molecule was persisting and that it could be involved in driving kind of this post-infectious illness. And so one of the major things that we wanted to ask was, is this a unique property of the Borrelia burgdorferi peptidoglycan? Or is this something that all peptidoglycan does and we've just never really realized it before? And so we did a series of studies over several years to, I think, pretty convincingly demonstrate that it is a specific property of the Lyme disease peptidoglycan, and it is important in the chemical properties of the Borrelia burgdorferi peptidoglycan are what's driving this persistence. But the other kind of question is, okay, well, the chemical properties are unusual. It's driving the persistence. How is it that our body's responding to this molecule? Because as I mentioned, this molecule is known to be pro-inflammatory in other systems. When we administer it into an animal model system, those animals get arthritis. So we know that it's inflammatory, but are there any unusual properties about the Borrelia burgdorferi peptidoglycan that kind of tweak the human immune response? And so we did a series of different proteomics and transcriptomic studies, and these are largely quantitative, but we don't obviously understand any aspect of the mechanism yet. But what we are starting to find is that there are some unusual overlaps and parallels in terms of how the response occurs to this particular molecule as it relates to other post-infectious diseases such as long COVID. And then if we take human immune cells and we basically feed human immune cells peptidoglycan from all different sorts of bacteria, there's a unique signature that the Lyme disease peptidoglycan creates. And that signature, in terms of the pathways involved and the metabolic processes that are involved, would, in theory, could really create a response of chronic fatigue. It was hitting all of the oxidative phosphorylation pathways and all these things that we know are important for energy metabolism. And those responses were the unique responses that only the Lyme disease peptidoglycan did. Again, these are ex vivo or in vitro and not really in a true animal or human system in many of these instances. And so we still need a lot of mechanistic detail to understand what's going on. But it really does seem like we're starting to kind of continue to stumble across this paradigm that this persistent molecule could really be involved in driving the symptoms even after the infection is gone. And then the last thing that I would say to that is that we also know that it's only a subset of patients that experience these long symptoms and chronic symptoms. And we're starting to work on this. And what we believe so far, and it still needs to be critically tested, is that is because there's only a subset of patients that have a hyper response to this molecule. So basically, it's coming back to the individual human genetic component, and that it is likely that there is a subset of patients that are really continually responding. And if they were to just ignore the molecule, they'd be fine. And because the molecule is an inert molecule, it's not causing anything. It's how your body's responding to it. And so we think that the subset of patients that experience some of these longer symptoms and longer courses of, say, Lyme arthritis may be because of how they're responding to that molecule. And it comes back to their own genetic variabilities.
So maybe someday there could be a test to find out if you're one of those folks who could be sensitive to Lyme disease.
We certainly hope so. That's something that we're very much thinking about. And these types of of tests exist for not necessarily many bacterial infections, but for many other types of disorders and diseases. So we don't think that is out of the realm of possibility at all. We think that's very well within reach, I think.
A lot of your colleagues and peers have called these two papers groundbreaking. How do you see them changing the landscape of Lyme disease research, treatment, and hope for years to come?
I think that, again, we're really just starting to scratch the surface. The field and the research and the science is starting to catch up. I should say that I really do think that the explosion of long COVID is really providing a voice for patients that have been experiencing long-term complications associated with Lyme disease. They're the original long COVID. This has been around for a long time. And obviously, it was the magnitude of COVID, the number of patients that got ill, and only that small subset that got long COVID. But there were so many of them that it's just basically a numbers game, of course. And so that, I think, is shaping how physicians and basic scientists like me think about these types of post-infectious sequelae or post-acute infectious sequelae and how they can shape the immune system, be it positive and obviously negative. And so it's my hope that we'll continue to think outside the box in terms of our understanding of these systems and allow us to put aside what we think and ask the fundamental basic science questions without any a priori thoughts about what we may or may not get and just follow the science. I mean, that is exactly what the folks in the lab are going to continue to do, and we'll go where it leads. And I think that's something that any science field, and particularly these days, really needs to do, is to continue to do the strong, very well-controlled, well-vetted science and see what it tells you.
Well, this is very exciting news, not only for those who are struggling with these chronic conditions from Lyme disease, but the 500,000 people diagnosed every year. So thank you so much for explaining this work so clearly and for all that you're doing. And welcome again to Northwestern.
Thanks so much for having me.
Thanks for listening. Please click the bell to receive notifications about our latest episodes and follow us on social media at NU Feinberg Med to stay up to date with our latest research findings.